Our next speaker is Dr. Doug Kane. Dr. Kane is a pulmonary physician who worked for many years in the Tennessee Upper Cumberland area. He recently wrote and published his first medical thriller, Broken Cure. Please welcome Dr. Kane as he speaks to us about the virus within us.
Can you hear me?
Yes, we can.
Okay. Good morning. I want to talk to you about what I find to be some interesting relationships between mankind and viruses. I thought this was a rather timely topic for us. The pandemic has stimulated unprecedented global research. The virus known as COVID-19 is in our daily thoughts. Viruses are not going to go away. I believe it's important for all of us to broaden our virology knowledge base. In this brief talk, I want to talk about a few interesting facts. In 1850, Louis Pasteur took diseased tobacco leaves, soaked them in water, and filtered the solution through a filter that trapped the bacteria, then put the filtrate on healthy tobacco plants. In a few days, they looked just like the diseased plants that he started with. So he made the conclusion that there is something smaller than bacteria that can only, in 1931, prior to that, the term virus was introduced to describe these little critters in 1898 by Martinez Pazernik. In the 1950s, Watson and Crick described the structure and function of DNA. So, are viruses alive? This has been an interesting question that's been debated over the years. First, let's ask ourselves, what is life? It's the condition that distinguishes animals and plants from inorganic matter, including the capacity for growth, reproduction, functional activity, and continual change preceding death. Others would add highly organized levels of structure, maintain homeostasis, respond to stimuli, and adapt to our environment. So let's look at a checklist. Do they maintain homeostasis? No, they don't. Are they highly organized? Yes, they are. Do they reproduce? Well, not by themselves. They've got to be within a host cell. Do they grow? No. They use energy, not their own. Do they respond to stimuli? The only thing they can do that would suggest this is that they do bind to cell receptors. Otherwise, all they can do is deteriorate in response to stimuli. Do they adapt to their environment? They certainly do. They mutate and evolve similar to other species. So by strict criteria, they're not really alive. But the general consensus is sort of, kinda, they're like bioandroids, which is probably the best description. So that leaves us with the following three entities: the living, the non-living, and viruses. What about structure? That's not a very clear image. That shows a collection of viruses which have very interesting and unusual shapes. All of their design is geared towards one function: hooking up to a cell and getting inside of it. COVID-19 is an enveloped RNA virus. The average viral size is 100 nanometers. To put that in perspective, the average type, the alveolar type 1 cell is 25,000 nanometers. So there can be thousands of virions in each cell. Looking at the structure, you see the spike protein, which you've probably heard about because that is a major target of 2 of the current vaccines. Inside the pink swirly, the pink coil is the RNA. The main route of infection with COVID-19 is through inhalation. This slide shows the replication cycle of coronavirus. Up to the left, you will see the virus attaching to the ACE2 receptor. It is taken into the cell. Once in the cell, it releases its RNA. This RNA is replicated using host enzymes. The viral RNA is also translated into structural proteins. These proteins are packaged into the envelope structure. The replicated RNA strands are then combined with the structural form, and you have a new baby virus, which bursts out of the cell. Since there's been a lot of talk about the ACE2 receptor and the use of the antihypertensive drugs, angiotensin converting enzyme inhibitors and angiotensin receptor blocker drugs, I thought I'd mention a little bit about this. Angiotensin is derived from renin, which comes from the kidney. It is converted to angiotensin 2 by the angiotensin converting enzyme. The angiotensin 2 binds to 2 different cell receptors, the ACE2 receptor and the angiotensin type 1 receptor. The effect of this and the molecules that result counterbalance each other. Generally speaking, the effects are positive. However, with coronavirus and other infections, they get a little bit out of proportion. The concern was that there would be upregulation of the ACE2 receptor, which would allow more virions to bind and worsen infection. In fact, there has been a lot of research done which has shown that it does not matter if you're on an ACE inhibitor or an ARB drug. It doesn't harm or hurt the individual sick with coronavirus. We indeed have a strange relationship with viruses. I want to just get into this a little bit because I personally find it fascinating. There are involved in 3 competing theories, discussing where viruses came from: the progressive, the regressive, and the virus first. The progressive theory is that a piece of eukaryotic DNA, eukaryotes, as you remember, are cellular, cells with organized nucleus like plants and animals. A piece of eukaryotic DNA transported from the nucleus, and which exits the cell within an exosome. An exosome is a membrane which is felt to eventually evolve into the virus's envelope. Over millions of years, it is thought that these bits of DNA that were within the exosome became more and more refined and led to the development of mature viruses that we know of today. The regressive theory suggests that viruses evolved from bacterial intracellular parasites. Chlamydia and rickettsia are examples. and that eventually evolved into structures containing only the genes required to replicate. The virus first theory is getting more attention recently. This suggests that viruses existed in the pre-cellular world before plants and animals as self-replicating units that gradually evolved into more complicated organisms. We generally think of viruses as causing infections and making us sick. In fact, some just hang out, fool our immune system, some wreak havoc intermittently, and some promote the development of various cancers known as oncoviruses. I find this to be rather spooky, and that is that approximately 10 different viruses have the ability to integrate into our DNA. What has been discovered over the last 20 years or so is that these viruses do actually leave behind bits of their genetic information. Fortunately, these segments are inactivated. At times, there's actually an exchange of genetic sequences, and at times, viral genes are stolen. Influenza is known to steal parts of our genetic code, which enhances their own replication. In addition, they can dysregulate host genes, which is part of their relationship with the development of cancer. This picture shows the life cycle of a retrovirus, and I'll run through this quickly just to give you a picture of how they insert into our DNA. Similar to coronavirus, the virus will bind to the cell receptor. It'll be taken into the cell where it spills its guts, which is RNA. This is transcribed in a reverse fashion, which produces DNA. This DNA is then taken into our nucleus, where it inserts into our genome and stays there. Meanwhile, it is transcribed. The viral RNAs leave our nucleus, they replicate. Other RNA will be translated into viral structural proteins. The two are magically put together, and a new virus leaves the cell by budding. There are numerous malignancies that have been associated with viruses. Many of these you've heard of, including Burkitt's lymphoma, liver cancer associated with hepatitis B and hepatitis C, herpes virus and Kaposi's sarcoma, which is primary in the immunocompromised. Human papillomavirus is associated with cancers of the head and neck, anal, oral, pharyngeal, and penile cancers. This retrovirus, the HTLV, is associated with adult T-cell leukemia. Merkel cell polyomavirus is associated with skin cancer and Merkel cell carcinoma. Over time, it's also been discovered that viruses are associated with numerous chronic diseases. It is believed that in time, we will probably be able to tie more chronic ailments to viral genetic insertions. So what's our relationship with them? It is felt that viruses evolved somewhere between 1.5 and 3.5 billion years ago. We and viruses have clearly co-evolved. One theory that has reasonable supportive science behind it is the development of the placenta. The placenta is what led to our ability to deliver live fetuses rather than eggs. Retrovirus sequences play a critical role in human embryologic development. Within the last, I believe, 7 years, several thousand retrovirus sequences have been found to be turned on when our embryo begins to develop. Viral genes appear to have aided in the development of our immune response and the production of critical enzymes such as amylase. Bacteria, fossil records show that bacteria date back approximately 3 billion years. It's estimated that viruses may have originated, as I said, 1.5 to 3.5 billion years ago. Direct sequencing of a 100,000-year-old Neanderthal DNA has revealed numerous bits of viral genome, affording the determination of mutation rate, which can then be backdated to give us an estimate of our coevolution. Retroviral RNA produces the protein Syncytium, which is the protein that promotes cell fusion, which is felt to have led to the development of our placenta. HERV, that stands for human endogenous retrovirus components. They have been inserted into the animal genes or inactivated. Several years ago, researchers at the Singapore Genome Institute reported that a portion of these retroviral genes are activated upon formation of the human embryo and play an important role in directing gene expression in addition to protecting the embryo from other viral infections. In summary, this lecture is ending rather quickly. The other one will be quite a bit longer. In summary, what I find most astounding is that it has been determined that viral genes make up a whopping 8% of the human genome. Endogenous viral elements, that's gene components, not complete active genes, have been found to make up approximately 42% of the human genome. And thus, in a way, we are virus. A few words about COVID-19. Currently, there is no evidence to suggest that it does integrate into our genome. There is no evidence suggesting that any of the coronaviruses are linked to cancer. It is not known to cause any secondary diseases such as the ones I listed previously. However, as you know, it can cause potentially chronic organ dysfunction, even in asymptomatic patients. There's been a lot of talk about COVID-19 and mutation. Its mutation rate is about two per month, which is a bit slower than influenza. The term mutation is often misunderstood. It is not synonymous with more dangerous. Helping means helping the virus propagate, which also does not equate with pathogenicity. Killing a host quickly is not good for the virus because it has less time to spread. COVID-19 is particularly successful because it has a very high transmission rate and spreading is vastly promoted by asymptomatic carriers. This whole mess started when a mutation allowed it to jump from bats to humans. In fact, one of COVID-19's mutations unfortunately falls into the helpful category for the virus. It is known as the 614G mutation. It resulted in a change in the spike protein that increases the virus's ability to bind to cells, replicate, and jump to neighboring cells. It first emerged in late February and is now the dominant strain. Now, this is an example of a mutation that has not had a major effect on pathogenicity in terms of severe symptoms or death. In summary, viruses are amazing, dangerous, and helpful. They are a part of us forever. Thank you.
Hello, does anyone have questions for Dr. Kane? Dr. Kane, I don't see any. We appreciate you and appreciate your presentation. And we will see Dr. Kane again after our break at 10 o'clock. Thank you.
Thank you very much.